It's no secret that DNA is the backbone of all organisms. To make new cells, we need more DNA. But how are we going to do this? We do it through DNA replication. For eukaryotic organisms like us, replication is located in the nucleus of the cell during interphase of the cell cycle. Before we get on to how DNA is replicated, let's talk about the directionality. DNA is anti-parallel and complementary, meaning that the strands of DNA run in opposite directions. One end of the template DNA strand is 5', which indicates the beginning of that strand with the phosphate group. The other end is 3', which is the only location on which we can add nucleotides to build the new strand of DNA. Complementary refers to the base pairing. If one nucleotide base pair is A or G, the corresponding base must be T or C, respectively. So let's get on to how DNA replicates. DNA is a double helix, meaning that there are two strands that will be used as templates for replication. We first use an enzyme helicase to break these hydrogen bonds so we can get these strands separated, creating a replication fork. Like hair clips, single-stranded binding proteins will attach to each strand so they don't come together. Topoisomerase prevents supercoiling tension. Now that this helix is open, it's time to start replicating. An enzyme called primase will first put down an RNA primer to signal to DNA polymerase 3 where to place the next DNA nucleotides. After DNA polymerase 3 puts down their complementary nucleotides, DNA polymerase 1 will remove the original RNA primer. To fill in that gap, an enzyme called ligase will put down the missing nucleotides. Since this top template strand runs from 3' to 5', the new anti-parallel DNA strand must run from 5 to 3, meaning that it will be continuously built towards the opening of the replication fork. This is called the leading strand. However, the bottom strand will not be made continuously, meaning it's the lagging strand. This is because the bottom template strand runs from 5 to 3', which means its complementary strand runs anti-parallel from 3' to 5'. The opposite direction of the replication fork. Thus, DNA polymerase can only make the strands in short sections called Okazaki fragments. First, each section still begins with the placing down of the RNA primer by primase. DNA polymerase 3 will make a short fragment of its complementary DNA. Similar to leapfrog, another primer is placed further down the lagging strand to signal DNA polymerase 3 and overall create a cycle of replication. After these fragments are placed, DNA polymerase 1 will remove the RNA primer and ligase will connect the two until the entire DNA strand is replicated. After replication is completed, you get two exact copies of DNA strands, which can either go into new cells for growth and repair or be turned into mRNA to make proteins.